In this slide, I've tried to highlight the differences between helical structures versus sheets. Under helical structures, we have three types of helices: alpha helix, beta helix, and 310 helix. Alpha helix is stabilized by hydrogen bonding. It's the most stable form found in proteins, as the strength of the hydrogen bond is around 4.5 kilocalorie per mole. If you look at the helix here, you can see that the around 3.6 amino acids per turn is present in case of alpha helix, wherein the torsion angles phi and psi, they are rotated to 100 degree. In order to form alpha helix, we need around 13 atoms to be present in the ring. Then we have beta helix. The strength of the hydrogen bond formed is around 0.5 kilocalorie per mole. We require around 10 to 13 atoms in the ring to form a beta helix. Finally, we have 310 helix, which is found in 15% of cases. The turn, the number of amino acids present per turn is around 3. The angles, that is phi and psi angles, they are rotated to 120 degree. The strength of hydrogen bond formed is around 1 kilocalorie per mole. We require around 10 atoms in the ring to form 310 helix. Now, one important feature of alpha helix is that they are amphiphilic in nature, as they have equal distribution of both hydrophobic and hydrophilic groups. Generally, hydrophilic groups, they tend to face towards the interior of the helix. There is an amino acid called as proline, which is called as structure breaker, that is helical structure breaker. This amino acid has a ring structure which does not facilitate the free rotation of phi bond. As a result, it does not aid in formation of the helix. Moving to beta sheet, beta pleated sheet can be of two types. You have anti-parallel beta sheets, okay, and the parallel beta sheets. In case of anti-parallel beta sheets, you can see here in the diagram, nitrogen, hydrogen, and oxygen, they lie in the same plane or same line to form hydrogen bonds. In case of parallel beta sheets, you see that NH and O, it does not lie in the same plane but at an angle. So this basically is the major difference between anti-parallel beta sheets and parallel beta sheets. Because of this, you see that anti-parallel beta sheets, they are more stable than the parallel beta sheets. In general, if you look at proteins in beta form, that is extended beta sheet form, they are much more stable than alpha helical proteins. If proteins have high amount of beta structures, they tend to have higher denaturation temperatures. Examples, we have already seen lysozyme, right? Lysozyme has a very high temperature of denaturation. We also have beta lactoglobulin when we talk about food proteins, which contain about 51% of beta sheets, meaning it has a high denaturation temperature. An example of a protein which has a helical structure would be probably casein, found in milk. Thank you for staying with me through the video. In case you have any queries, please feel free to put them in the comment section and I'll get back at the earliest.